And I guess I should, since no one's going to introduce me, I'll introduce myself. My name is Steve Mayo, and I am the chair of the Division of Biology and Biological Engineering at Caltech, and I will be the moderator for today's session. I want to start by just saying that we hope this is an interactive session and that the audience will participate in providing questions, comments, or perspectives on the topics that we're going to be talking about. We're going to try to cover three things. The first is thinking about the ways that universities can be venues for new approaches to bioscience. The second is more patient-oriented and thinking about the university role in things like clinical trials and technology transfer. And the third topic, if we have time to get to it, will focus on challenges for the future, including things like the impact of the sequester on bioscience funding in the U.S. So to start, let me introduce the other panelists. On my right, far right, is David Baltimore, President Emeritus and the Robert Andrews Millikan Professor of Biology at Caltech and winner of the 1975 Nobel Prize in Medicine. Dr. Baltimore is also director of the Joint Center for Translational Medicine, which is a joint project between Caltech and UCLA. Joel Burdick is the Richard L. and Dorothy M. Hyman Professor of Mechanical Engineering and Bioengineering at Caltech. Dr. Burdick has a background in robotics and over the past decade has been working with a team of researchers on the development of neural prosthetics. James Economou is the Vice Chancellor for Research at UCLA and the Beaumont Professor of Surgery and Chief of the Division of Surgical Oncology at the David Geffen School of Medicine at UCLA. He's also played a significant role in the development for the Joint Center for Translational Medicine. Michael Quick is the Executive Vice Provost for Research and Professor of Biological Sciences at USC. Dr. Quick previously served as the Director of the Neuroscience Graduate Program and Vice Dean for Research at USC. And on my far left is Owen Witte. He's the Director of the Broad Stem Cell Research Center and the President's Chair in Developmental Immunology, Microbiology, Immunology, and Molecular Genetics at the David Geffen School of Medicine at UCLA. He's also David's co-director of the Joint Center for Translational Medicine. So with that introduction, let me just sort of lay out what we want to talk about for the first section. So it's clear that the century will actually provide great opportunities in biology and the biological sciences and provide a great venue for translating those activities into many applications, including healthcare. One of the things that we're interested in doing at Caltech in particular is thinking about how we can actually integrate biology with engineering approaches. And most recently, we've actually created a new division, the Division of Biology and Biological Engineering, which is an attempt to actually integrate more explicitly sort of basic fundamental research in biology with translational activities. And so from our perspective, biological engineering really is a broad way to think about translational activities, including translational medicine. So when I think about biological engineering, I think of it somewhat differently than bioengineering. So in traditional bioengineering, for example, the substrate on which you operate on might be more physical, like silicon, in terms of making a medical device. Biological engineering uses biology as a substrate, molecules, networks of molecules, cells, tissues, et cetera. And so we're thinking about the idea of providing sort of intellectual space for integrating these sort of activities, bringing concepts of engineering into the biological sciences with the explicit objective of generating engineered biological materials. And to start, I might ask David Baltimore to describe a project that he's working on in an area called engineering immunity. David.
Yeah, I actually talked about this a little bit in an earlier session today, but I'm not sure that there's an overlap. And so let me go back. We are trying to use genes to improve the functioning of the immune system. Now, there are lots of genes that we know about, and all of them are fodder for application, for engineering application. But the question is, if you have a gene, what do you do with it? And viruses solve that problem for us because viruses, certain viruses, can carry genes into cells and actually can make those genes parts of our chromosomes. Or they can set up a little factory in the cell to turn out the products from the gene that's brought in. So viruses are an incredible substrate for what we generally call genetic engineering. The engineering of a cell or an organism to do something different as a consequence of new genetic material that's been brought in. So it's all about DNA. DNA is an incredible molecule because it establishes a code, and that code makes many things biological effectively digital objects. And so we deal with it as chemistry because DNA is a chemical, but we think about it in terms of information transfer. So when we're designing molecules that are going to change the properties of cells, we think of it as a combination of digital elements. We've made some vectors recently that allow us to express antibodies in muscle cells. Now, muscle cells don't ordinarily make antibodies. So we've got to put in a gene for, actually, a couple of genes for antibody production. But that's easy because there's a virus, adeno-associated virus, which is all set up to do this. And all you've got to do is take out the virus's genes. Now you've gotten rid of any pathology. This is now just a plain old vanilla vector, as we call it. And we put in the genes we want. And we put in those genes with all sorts of controlling elements to make sure that they're made, the products are made in large amounts, if we want large amounts, and that everything is set up consistent with the principles of molecular biology that have evolved over, that we've learned about over the last few decades. And that takes a little bit of an SV40 virus, it takes a little bit of a cytomegalovirus, it takes a little bit of the adeno-associated virus itself, as well as the genes for antibody. And you put all those together, and this is true biological engineering, because what we're doing is taking our elements from wherever we can find them and combining them to allow a biological change. So what do we do? What are we trying to do biologically? What we're trying to do is to get muscle to produce an antibody that will prevent HIV infection. So we're actually turning viruses against viruses. Now, that may sound like we're making a vaccine, but it isn't. Because vaccines are the product of the body's immune system. And we're going around the immune system. We're actually turning muscle into an organ of immune function. And when we do that with a single or a small number of antibodies, and they're very carefully chosen. So where do we get them from? Well, blessedly, there have been antibody hunters out there looking for antibodies that prevent HIV infection. And in the last, particularly in the last 5 years, a number of laboratories have found terrific antibodies of this sort. Antibodies that work at relatively low concentrations, and particularly antibodies that will work no matter what strain of HIV you have. So you've all heard about HIV's protein nature. It can constantly change its genetic structure. It can elude most attempts to control it. Well, these antibodies are finally, a step ahead of HIV. And because the initiation of an HIV infection is something that happens from only really a single particle or maybe a couple of particles being transmitted from an infected person to an uninfected person, the actual job of preventing an HIV infection is a pretty easy job. It's easier than most viruses. Most viruses, thousands of viruses get in from an initial infection. And so you've got a much bigger job if you really want to prevent the initial take of the virus in the body. So these combinations of genes and vectors and little bits of molecular biology being done by elements from various viruses are what I see as biological engineering. And I really believe it is part of the future of medicine. We're going to make these things, we're going to make them safely, we're going to know that they are safe because we're going to have tested them in large numbers of people. At the moment, it's only in large numbers of animals. And we believe that there is a possibility that we can literally prevent the transmission of HIV with this engineered virus. So that's an example. I don't think I should go any more into, although we're doing many other things in the realm of engineering immunity. And I think Owen will talk about how we have as a collaboration the clinical strengths of UCLA with the biological engineering strengths of Caltech.
Just one thing to add to that story is that, you know, there's also an opportunity to really engineer the antibodies themselves and not just rely on antibodies that one would discover naturally.
Right.
Owen, do you want to add to that?
I could just add a little bit on a separate project, which is to modify cells of the immune system or hematopoietic stem cells that will turn into cells of the immune system that become cytotoxic T cells. Again, a virus is used to introduce a therapeutic principle, in this case a T cell receptor gene, although one could imagine other genes such as chimeric antigen receptors that are also being used currently in different institutions. But each of these ideas can be modeled, I think, quite effectively in animal models, test the concept, build the various components. But one of the rate-limiting steps is to how to actually cross that large chasm and get these therapeutic concepts into people to test their validity, not in a mouse or another animal species, but in a human. And I think that's really been one of the interesting changes that I've seen in the academic climate over the last decade or so, is the realization that we don't have to always hand it off to the biotech or pharma industry, that we can do a great deal to prove the value of these concepts in humans, totally within an academic setting. And David and Jim and I have all been involved in this Engineered Immunity Consortium, which involves Caltech, UCLA, and some other sites, including somebody from USC, and which is good, and I hope. What we've done is that we've put together a group of people who have all of the standard academic purview and influence and interests and so on. And, you know, academia is essentially an egocentrically driven enterprise, like any discovery enterprise. But we put ourselves together realizing that separately we can't get that far, but together we can. And it's been amazing to me that in a period of less than 7 years, it went from the certain concepts that grew out of David's lab and Jim's work and other colleagues like Tony Ribas, and have put together constructs, materials, GMP manufacturing, all the technical processes within an academic environment, and done clinical trials with 14, 15 patients to date to test out this therapeutic principle of engineered T lymphocytes for the treatment of certain types of cancer, in particular melanoma. And that could not have happened without a really sea change in the way people think about how academic research can be construed and how it can be supported, which I think is an issue we'll come to. But I think it takes different kinds of grant mechanisms, different kinds of foundation support, and different kinds of institutional support to do this type of work.
Thank you. So USC has recently, relatively recently, started a program that integrates engineering more directly into biomedical activities. So maybe, Michael, you can talk about that.
Yeah, sure, Steve. Happy to. Yeah, so as Owen mentioned, you know, one of the the big issues we have is trying to narrow that gap and the speed at which we want to move things from sort of the sort of basic science side to application. And you know, thinking about what are the major drivers of trying to get things sped up a little bit. And you know, we've been at USC, we've been working on a number of just different approaches to that. So I don't want to dive into the depths at which the science is being talked at here, but more just talk about strategies we think have been helpful in sort of getting the biology and the science together. Because some of it has to do with cultures of how different disciplines approach issues. And it never hurts to take these folks and whether they're basic scientists, physicians, or engineers, and actually do the simple things like putting them in the same building so they bump into each other. It's an amazing thing to watch happen. And when you keep them away, sometimes it really slows down the opportunities to have accomplishment. And so we are now trying to create, as Steve mentioned, structures that integrate the sciences and the engineering, buildings that integrate sciences and engineering. There have been some amazing technological advances that bridge between the biological sciences, for example, and engineering. But let me just throw out one that I think is possibly the most important. That is the espresso machine. And I don't think one should downplay the importance of things like that bringing people together around those types of things have profound impacts on the way we do science and the way we get science done. And moving from sort of discipline-based approaches to problem-based approaches, to instead of defining things as an engineering problem or as a science problem, to define them as here's a specific problem of solving HIV. And by reframing the question, we've seen a lot of advance come about. The specific issue that Steve raised is a program that we started at USC called the HTE program, the Health Technology and Engineering program. A faculty member jointly appointed between neurology and biomedical engineering, Terry Sanger, heads this up. He, and it's based on a model that Harvard and MIT have done for a while. It really is aimed at another thing I think we need to do more of if we are going to shorten this gap between the basic sciences, biology, and engineering, is to get these cultures together as speaking the same language. They all, most use English, but oftentimes they don't realize they're actually talking about the same problem. And so what Terry has done is he has a program in which engineering PhD students go to our health sciences campus and spend 6 months to a year working with an MD student in the clinic. So the MD student is doing rounds, the PhD engineering student goes right along with them. And they start to learn the language of medicine and how one approaches patients, how one as a physician has to think about approaching patients and what those problems are. And they try during that time to define a problem that needs to be solved, a better instrument, whatever that may be. Then the engineering PhD student and the MD student go into the laboratory to create that device or to work on that problem. And they spend 6 months to a year working on that. And so what you get at the end is an engineer that knows a little bit about medicine and a future physician who knows a little bit about engineering. We've only just started this, so it's hard to see what the long-term outcome would be. The hope would be maybe this is a different model to what has traditionally been done in sort of MD, PhD programs where students do their clinical training and their basic science training together. Maybe this is a different angle at, once again, speeding up this interface between the gap between the basic sciences and engineering. So that's the HTE program at USC.
So you mentioned MD, PhD programs. I just wanted to also mention, just for additional interactions between these three institutions, is that there actually is a joint MD, PhD program between both UCLA and Caltech and USC and Caltech, which I think is really tried, again, to bridge this gap of basic biological research and being more translational. It would be great if someone from the audience has a question or remark to make. I don't want to just have us all sitting up here as a bunch of talking heads. Any comments, please? Should I ask someone? Please, go ahead.
My name is Nick. I'm actually a graduate of the UCLA Caltech MD PhD program, and I'm a postdoc with Peter Driven and also a resident at UCLA. Dr. Witte, you mentioned that there's been a change last 5-7 years whereby if you have something in the lab and you wanted to try it first in human study or something, that it's more likely you're able to do that now than before. What are the funding situation for those types of things? Because that's not a typical R01 style thing to do. No,
it's just the opposite of a typical R01 type grant. In fact, I don't think there is a current standard model for it, and we had to, in a sense, evolve the funding by being an interested group of people. Actually, Jim Economou was one of the key people in this enterprise of getting us funded. We went to various private foundations, applied as a group for programmatic style grants. Eventually, several foundations supported our work early on, as well as philanthropic money that came in through the stem cell center that I had, and other monies that David Baltimore had available. But the key was really when we had reached a critical mass and could apply for a programmatic grant, a PO1 grant from the National Institutes of Health. So that helped sustain the program to move this therapeutic concept into a trial. And then finally, recently have funding from the California Institute for Regenerative Medicine, which will provide about $20-something million over a 5-year period for some of the actual conduct of the clinical trial, production of the materials that we need for the trial, et cetera. So there's really there are mechanisms out there, but what was needed was to get a sufficient number of people involved and build a team to be competitive for those resources.
Can you maybe come and use the microphone, please?
Sorry. I was going to ask a question about, I guess, the way science funding is changing in terms of maybe NIH having more cooperative agreements than R01 grants and also these program projects and so on. And I just wanted to know what the panel thought about that evolution, because that's changing the way basic science is being done at the very least. So.
Anyone want to comment on that?
I think maybe we should turn to Francis.
Francis Collins comment on it?
We have perhaps the ultimate.
I was going to get to listen to one at least get paneled. So if you look at the actual numbers, the percentage of NIH funding for basic versus clinical science, clinical applications has not changed substantially over the last 20 years. It's about 53% basic and about 47% clinical. Now, some people will argue about what's your definition of basic, and so if you broke it down into basic, which is people, for instance, studying a model organism with no intention to connect that to disease versus people who have, as part of their grant application, some reference to interest in a particular disease state, then that would be about half and half. That basic part would break down. But in terms of the mechanisms, the R01 continues to be the mainstay of investigator-initiated efforts. But it is certainly true that if you're trying to undertake a team-based effort, and science is increasingly team-based, the opportunity to use other mechanisms, you mentioned the PO1, but certainly cooperative agreements can also be very helpful, especially if you need sort of a steering committee and you need to be able to have some staff involvement as well to make sure that projects are staying on track. I don't know off the top of my head what the change in numbers. It's been subtle, frankly. The R01 proportion has not been greatly diminished by the focus in some places on team science. I think there's more of an urban legend about that than is actually the case. And of course, everything is tight right now, and unforgivably tight right now, which means that anybody who's trying to get funded by any mechanism sees that it's harder than it's ever been and assumes it must be because somebody else is having an easier ride. And let me tell you, nobody's having an easy ride right now, and we have got to get this turned around.
Thank you. Maybe we'll come back to questions like that in a moment as well. So maybe we should transition to the second topic, which is sort of patient-oriented and thinking about ways that universities can partner with industry and to be more entrepreneurial, tech transfer-oriented type activities. So Joel Burdick, I think, will say something about his robotic work and his efforts to engage the tech transfer engines.
I'm the token engineer on the panel. And so I've been involved with a great collaboration with a team over at UCLA doing animal work, and now at University of Louisville doing human work on spinal cord injury. If we have time later, I'll show you some fun videos. But as we went through the animal work and got to the human work, which is started funding by NIH, we reached a point where we wanted to expand our trials. And unfortunately, that size of what we needed to do was difficult to fund under a normal NIH sort of mechanism. So we started going down two paths. So one is we were contacting a number of large companies in the medical device area to see if we could team with them, and we were somewhat surprised by the answer. They were like, no, this market's a little bit too small and it's too risky right now. But if you start a company and it's successful, we'll buy you. So this is really speaking to what's sort of coming later, which is increasingly what we see. It's a trend that's been going on for a decade, but I think increasingly now with the sort of bite on the healthcare coming from upcoming regulation changes and just the economy in general, companies, especially in the medical device side, I don't know pharma, are increasingly reluctant to put the risk capital out there to really advance both clinical trials as well as technology. So the way we've been doing it is by using NIH when we can. We're working with two great foundations in the spinal cord area. The Christopher and Dana Reeve Foundation is the premier group, and they're a fantastic group of people in terms of being helpful. And now also the Helmsley Foundation. We sort of went further, and we went back to the large companies, said, okay, we're further along. Are you ready to work with us? We've proven the science and proven the technology, and they're like, no, it's still too risky, but we'll buy you out if you come back later. We've actually started a little company. I'm a reluctant entrepreneur. I've tried it before, and I'm awful at it. But in some sense, we've not been able to get the large companies to really do things. Their model is, you go out and get the risk capital somewhere else, and then if you're successful, that takes the risk off the table, we'll come in and buy you out. And I see that, at least on the medical device side, increasingly the model has actually almost become the written policy of the large companies. And especially if it's not a blockbuster market, that's kind of their current standard of practice.
Why don't you, we have time. I think maybe seeing the videos would be interesting. If we could
if we could get video, actually we'll go through all, I just have 3 slides. 3 slides, number 1, please. So those of you who don't know much about the spinal cord market, there's about 1 million people in the US with spinal cord injury. About 400,000 are severely injured, and about half of those are what we call spinal complete, which means they have no motor commands or sensation below the level of their injury. What you may not know is the medical burden that places on both the patients as well as our economy. So it's about a $10 billion a year sort of extra healthcare costs because the average spinal cord patient will have $2 to $4 million of increased health care over their lifetime because of all the secondary complications. So if you go to the second slide, please. And so what we've been working on is through animal models and then through initial trials, if you could just click forward and play the video, is implanting specialized devices in the lower spinal cords of patients to be able to get them to move again. So this is our first patient. If you go, I'm sorry, go back 1 slide. Our first patient, Rob Summers here, was completely paralyzed below the level of his armpits. And so you have to go back and play the previous video, please. Thank you. And so we implanted specialized devices in his lower spine to be able to trick the spinal cord below the level of the injury to be able to think it's standing at a cocktail party. So this is Rob, where we're now ramping up and turning on the implanted electrode. And so now he wiggles to indicate to us, I'm actually ready to go now. And so he was able to then sort of stand independently and get great gains in his healthcare side effects from this technology. So if you go to the next slide in video also then, and then about 7 months afterwards, in an unexpected event for us, he started recovering voluntary motion due to the stimulation of this technology. And so he was actually able eventually to be able to step independently, again, in a very limited sort of clinical setting. The point I want to make of this one is that the technology, which is approved by the FDA at this point, does not allow us to stimulate him in a way that we know from our animal models will allow him to walk left and right. If you look closely here, he can move his right leg, but not his left leg. And so we can can switch it, but we can't optimize both. And unfortunately, we sat down and sort of penciled out what we need to do to get to that next step. And you know, it's a very expensive proposition, and the large companies feel it's just too high of a risk at this point to be able to do it. So we're struggling with foundations, and not only the R01 mechanism, we actually aren't using R01s, where we have a BRP, which supports the basic science. We now actually have a U01 grant, which actually is a great little program to help people translate technology. And so we're also looking at this technology for upper body paralysis as well. But to sort of echo what I heard down from Owen before, really us ivory tower academics have to become entrepreneurs. We have to go out and actually put together big teams, multiple funding strategies, and actually have to sort of jigger these things over time to be able to get to the ultimate goal.
Thank you. So Jim is going to have some comments on entrepreneurship in clinical trials and universities' role and things like that.
Thanks. So I'm the only practicing physician on the panel, and exactly 20 years ago, Ari Beldergren and I at UCLA conducted the first gene therapy trial in the western United States. It was a Herculean effort. We had to build all the infrastructure from scratch. We went to Washington, D.C. 3 times to go to the RAC. We went back and forth with our IRB over a dozen times. Now, actually, the IRB reports to me, but then it was a miserable experience.
Why don't you unpack those abbreviations? IRB, RAC.
Well, the Recombinant DNA Advisory Committee and our institutional review boards for clinical trials. Yeah.
And just maybe just say a second about what the IRB does and why they're critical.
So the IRBs are very important because, and since I actually, one of my responsibilities is overseeing all research involving humans and all research involving animals, there are many regulations that require us to ensure that we're following federal, state, institutional, moral, and ethical guidelines in conducting this research. So certainly when we were conducting this trial 20 years ago, it had to go through multiple rounds of review. The byproduct of that was, though, that I asked the dean if I could found our gene medicine program and to build this infrastructure and to build a culture of this kind of translational medicine. It really built, with the help of many others, one of the largest human gene medicine programs in the country that have enrolled now several hundred patients in over 45 trials. However, when we did this in the 1990s, many institutions had not made the necessary investment in building this kind of infrastructure. So I want to comment, make a few comments on clinical research. Now in this decade, a number of remarkable things have happened. We have these powerful tools that allow us to directly study human disease biology. Genome sequencing, we have large annotated biorepositories, functional imaging, electronic patient registries, and now this allows us to interrogate human subjects and to gain information that then informs iteratively improved clinical trials and informs basic science, whereas before it was mostly basic science informing clinical trials, which then ended up being almost proof of concept exercises. So there are about 4 ways that universities, and in particular academic medical centers, can accelerate biomedical research. The first is that we have to make large-scale investments in modern research engines to study, conduct clinical investigations in human subjects. And it has to have an emphasis on discovery at the patient level. NCATS, the National Center for Advancing Translational Science, was a remarkable sea change in how the NIH supported clinical research. In the past, we had these clinical research centers which were perhaps not functioning at the level that they should, and now we have 50 or 60 of these clinical translational science institutes that basically are required and mandated to interact with one another. I'll give you an example. Of the 5 UC biomedical complexes, Davis, San Francisco, Irvine, San Diego, and UCLA, all have CTSA grants. We now have integrated IRBs so that someone in San Diego can open a trial and someone at UC Davis can enroll a patient. We now have linked research data repositories, so we can link these de-identified patient data. And we also have biorepositories, patient samples that are linked. So that becomes the largest platform for clinical and translational research in the world. The second is that we need to develop mutually beneficial and respectful partnerships with pharma. Pharma no longer has the resources to do a discovery, to do a lot of the discovery, and they don't have the kinds of insights into human disease biology that we have, but they're really good at making drugs, and they really know how to deliver these and commercialize these. The third is that, and this has been alluded to by David and others, we need to recognize the importance of team science. These big problems can't be solved by one or two investigators. And I think that the team science effort that we enjoy with our colleagues at Caltech and USC, now numbering, I think at our last meeting we had 50 people at our 4-hour Saturday morning meeting where we reviewed data that included data from clinical trials, basic science where we're trying to figure out whether we're getting the biology correct. So it's team science where you have basic and translational and clinical scientists all focused on a goal to improve patient care, because that's basically what the ultimate outcome of biomedical research is, changing the care of patients through this process of discovery. This also is going to require us to kind of change the academic model that we have, which is kind of based in the 19th century, where we have departments and schools as opposed to disciplines. And it's also going to require us to change the academic reward system for team science members, to recognize that everyone participating in these teams has an important role and that we need them in order to advance to the proper goal. Finally, we need within university systems to create pathways for entrepreneurship so we can deliver these discoveries and new treatments to society, and we can do these quickly. So I'm just going to show you one slide, but I'd like slide number 2. So one of the efforts that we've been making on our campus for the last couple of years is to create more of an entrepreneurial ecosystem for the delivery of discoveries and innovations from our faculty and students to society, which is what we ought to be doing. A traditional model has been one where a disclosure is made, there's a provisional patent, and the tech transfer office works as hard as possible to get the thing licensed before they start accumulating patent and legal expenses. And so what happens is a lot of stuff falls off the table. A lot of things that potentially have promise really don't get matured and moved out of the university. So this ecosystem really requires a coordinated effort across the campus. Business advisory boards at the domain level, not necessarily the school level, but basically at the fundamental scientific domain. Integrated educational programs at the undergraduate and graduate and professional level, on-campus incubators and technology platforms, a business decision-making approach which requires you to bring business people who are friends of the university onto the campus and to help make the key decisions in terms of what to move forward and how to develop it. Proof of concept grants, and all of these have the goal of keeping discoveries and startups within the university environment as long as possible, to mature them, to refine them, to prepare them to be launched into the private sector. And that will then increase the value transition point from a little further out of the university towards the university, so that we can capture some of the licensing and royalty revenue streams that can then be reinvested in academic programs in this constrained environment.
Thank you. I think it would be really interesting to hear from some audience members, particularly those who are associated with, have been associated with tech transfer, or who are part of so-called big pharma. I know there's at least one of them here. Maybe he will comment and say something about university industry partnerships. It's, Remy.
My name is Remy Broy. I'm representing Sanofi for the West Coast. And yes, we have a wonderful partnership with Caltech. We've been trying with you, Steve, we thought about what could be the optimal way to work together, helping you to do disruptive science, even before going to NIH. So that was the idea at start, and helping some of your scientists to develop new ideas. And then if something come out of it, we could then discuss a sponsor research agreement, or you can go back to an NIH funding. And so that was one of the original things that we've been developing together. I think that's one possibility. I think there are much more. I like the idea of having a medical school with you. That's something that I like, the concept of the cafe where people can be together. We also have developed a partnership with BioX, and the best restaurant of Stanford is within this unit, so you have everybody coming and discussing together, the scientists, the basic scientists, the MDs, the PharmD. I think that's another way to work together. Integrating a school is important for industry. We need to be able to develop translational medicine as well, and having as quickly as possible a proof of concept in human. So having a school of engineering with MD, developing biomarker, being able to follow a disease and being able to select a subpopulation is something that pharma is certainly looking at, and that's the way to success. Yeah.
Thanks, Remy. Any other comments? Yes, please.
Good afternoon. I'm Joe Boystack with Brightwaters Capital, and very active with respect to academic industry partnerships at several universities, including UCLA, Johns Hopkins, and Harvard. USC is also emerging on our screen as well. This is ground zero right here, what you gentlemen represent as far as innovation for the American economy and global impact. What we need here, and where there's a real breakdown, and we've had considerable discussion over the course of this conference, is access to capital. As you can put some wonderful paradigms up, structures, and Jim, what you've outlined here, I think is the model for how the academic industry is evolving. But without continuous access to large-scale capital, those plans will not fully manifest on that. So there are a couple of elements that at least I see as critical, having spent most of my career as an investment banker with several major Wall Street firms. And one is an environment that's open innovation, and it's not siloed, but it's open innovation, which really invites in a meaningful way industry and academic partnerships. Part of that includes shared risk agreements of how capital and risk and exposure occurs in developing this, knowing where each party can maximize its input and develop accordingly. The notion of Boston Scientific or J&J or whomever that was, Joel, that you were dealing with, say, go build a company and develop your entrepreneurial skills, is really an old notion, and it's destined for limited success. You should be focused on the research and bringing together then resources that can supplement you, not make you go out hat in hand and try to raise capital. So I think it's how do we begin to pool assets and look at new funding structures, because the financial ecosystem is ruptured. And we have a very effective NIH and their NCATS program. Angel funding has been episodic. The breakdown has occurred within the VC community, and that's not going to restore quickly or if ever to its original glory. The public markets are robust, and we're seeing how companies that can access public capital are doing, but the breakdown and where this whole thing will bottleneck is identifying new sources of venture funding. And again, it's not going to be single molecule companies with binary risk that will attract large-scale capital. Rather, how do we pool assets that come out of UCLA, out of USC, out of Caltech, around themes, and look at large-scale access that could perhaps become mutual funds or ETFs, and open up a whole new generation of investors to help enable and facilitate this. And there are sophisticated ways that you can layer risk to attract different types of investors. But I think that's got to be part of the calculus here, and very active in the discussions.
That's interesting. So do you think there's a role for venture capital directly invested on campuses?
Absolutely. But again, it's not a single company, single molecule approach. It's going to be around this pooled construct. We're seeing pharma starting to do that, as corporate venturing is beginning to eclipse some of the traditional venture funds. So, for example, Center for Therapeutic Innovation that Pfizer has sponsored, some of their collaborative initiatives up at UCSF, I think, is taking steps in that direction. But it's going to take more than the venture capital community alone, and it's going to require incremental sources of capital to really fund and fuel this.
Steve, could I say a word?
Please, David.
I'd like to understand this better, because my experience has been that it's absolutely imperative to focus on one thing at a time, or one thing by itself. And that if you start aggregating a lot of different things, none of them get enough money, and they either end up competing with each other for whatever funds are available, or they end up each one dying an unpleasant death. I don't see this shared model that you're suggesting, and I particularly don't see it in terms of the interests of investors in wanting to know what they're investing in, and where it's going to go, and how they're going to ultimately profit from it.
So I agree generally with the comment. It's not a potpourri, what I'm suggesting here, rather very themed type of pools that could get constructed. So it could be around a particular disease.
So you're talking about a platform as opposed to a particular, binary.
Platforms.
Yeah.
Platforms imply a certain, either delivery mechanism or something to that effect. But no, rather, let's look at a laddered portfolio of programs that may be in pancreatic cancer. It could be a combination of companion diagnostics along with the therapeutics. And begin to, so you can look historically at what risk profiles have been by therapeutic category, what the success rates have been based upon their approval processes. And you can begin to layer risk if you have a preclinical program along with a phase 1 or a phase 2 asset. Couple that with a companion diagnostic. This begins to create coherent pools that people can understand a theme and understand the risk profile.
That's interesting. If there are no other comments on that, maybe we can move to...
I'd like to make one comment, please.
Okay, please. Go ahead.
So I've worked in technology transfer, I've worked in pharma, and I'm a doctor as well. And also worked in finance. I get distracted frequently. I disagree with the gentleman that venture capital money is probably what we need more of. I think that the time investment horizon is incorrect for venture capital. It's not patient enough. I think that there is a fundamental difference when we talk about what type of science that we're doing. Are we doing basic science for the sake of science, or are we doing, as the HTE program tries to instill, science for an application? And so I think the way across this valley of death, which Joel Burdick was talking about, which is when a company has a great idea, but a larger company says, it's too risky, we're not going to give you money for that, come back later. I think the way across that is potentially a program which you mentioned Pfizer had laid out, the CETP program, where they have fundamentally invested upstream across the valley of death, reaching out for a reason, for a particular type of research, for something that plugs in to a therapeutic area in their portfolio that they're interested in because they're closest to the market. And so I think that's probably has the most promise to bridge that gap now. And I think it's up to gentlemen like you at the table here to reach across to people in pharma, medtech, Medtronic, for example, these types of companies, to try and understand how to put together a pool of funds that are dedicated to a particular type of research projects that then in the scientific universities, you can then say, we have this money for this reason. Do people want to come forward with research they're doing for this? And that can be shepherded through the technology transfer office as an agent to take the science, transferring out of the academic university and into the marketplace.
Thank you. Francis, please.
I just want to ask the assembled university experts here about an issue that certainly has caused a lot of vexation over the last 15 years, which is conflict of interest. As you know, a few fairly spectacular examples of investigators who were involved in relationships that involved substantial personal financial benefit but didn't disclose that, and potentially, therefore, seemed to have affected their scientific judgment, caused a great deal of distress and a need for NIH. My predecessor, especially Elias Zerhouni, spent a lot of his time trying to figure out what to do. So there are now new, much more rigorous financial conflict of interest guidelines, which require disclosure, require every university to have a system of reviewing those, deciding what's significant, and being able to make that available to members of the public who ask. When those were being put in place, there was a lot of anxiety about this was going to really end the ability of universities to work with companies. And we all tried to argue that that would not be the case because it's disclosure, and you're certainly encouraged to build those relationships, but there are limits in terms of personal financial benefits that can be tagged into them. I haven't heard anybody talk about that in this conversation. Should I assume that it's all fine now and there's no problem and everybody has got with the new program and it didn't turn out to be that onerous after all?
Well, Dr. Collins, unfortunately, also the Conflict of Interest Committee reports to me, and I'm also the Chief Integrity Officer at UCLA. So our philosophy has been that we would like our faculty to have as many potential conflicts as possible. We want them to engage in discovery-based research and scholarship. They're at our research university not only to teach, but also to deliver their discoveries to society. That's why they're there. And even if they don't make money, that's something that they're very passionate about. And to do that, they're going to have to engage the private sector sometimes in a meaningful fashion. So the Conflict of Interest Committee, in the last couple of years, I've encouraged them, and it's entirely a faculty-driven committee, but it's a committee that helps faculty identify and manage these conflicts as opposed to catching them or telling them they can't do something. And we even have a concierge service where a faculty member can come in and say, you know, I'd like to do this, this, and this, and they'll sit down and say, great, but this is how you should do it. And this will be basically within appropriate university and federal guidelines, and you won't be getting a letter from a senator in a couple of years. We've also found that a lot of the conflicts that exist on some of the university rules and regulations are circular, they're dead-ended, and they're sometimes self-defeating. But I think that having a culture on a campus where you're encouraging this, you're helping faculty to manage it. You're saying you probably should not do this, and you've got to disclose this, this, and this in these kinds of settings. That really, to a large part, I think, alleviates a lot of the concern on our campus. Now, we've also got all the paperwork that we've got to do, and I'll send you the bill for that a little bit later. But I think it's a different culture that we need to have.
Yeah, I would agree with that. I think in general, you know, it's clear that the new regulations generated a lot of angst among a lot of people, but at the end of the day, I think things are working quite well. To echo what Jim said at the end, I think the real sort of quantitative impact has really been driving compliance costs. But I think in terms of people, certainly at Caltech, engaging in an appropriate way in terms of being mindful of regulations and disclosing when appropriate, I think that's all working out very well.
Yeah, I would just say from down in the bottom of the trenches here. So certainly, as we talked about, as Charles mentioned before, there's always this valley of death, and that gap's getting bigger now as some of the large contributors are pulling back. And so the ability to do entrepreneurship is helping us to fill that gap as a necessary evil. And I think from my level, the universities have really engaged in that process quite nicely. And so it hasn't really caused many problems. In fact, I feel I'm a better investigator. We've had to go through online training courses and other things where we really understand the philosophy behind the conflict of interest and actually how carefully to structure things to avoid it.
Right. So we're right at the end of our hour. We have a couple minutes left. If there are any closing comments that someone might want to make over here, please. And then I think we'll have time for a short response.
Thank you. I'm a professor of cardiology at UCLA, NIH funded. One question I had for the panel and perhaps some other experts in the room is, one of the best bipartisan moves ever done by the United States, some people say, is the Bayh-Dole Act. And now the Stanford v. Roche decision of the Supreme Court was somewhat disappointing for many of us. What is the thought from the experts on the panel and anyone else in this room, and perhaps also from our industry expert who just spoke? Do you think that moving forward this would have a legal impact? Because that is very central to academic sort of industry partnerships.
I fear that we don't have time to dive into Bayh-Dole and the recent Supreme Court ruling. But if there's a 30-second thought on that, that would be. Sorry, I think we'll have to pass on that. Yes, you had a question, a comment?
Well, this might be equally as vexing, but I was just curious about what the change in the U.S. patent law from first to file to first to find to first to file has done in terms of universities and other investigators' interest in patenting discovery.
Well, I would say certainly at Caltech it probably has not had any impact. I know that the real sort of tension and drama was really in the industry. Does anyone else want to say anything about the first to file implications? I think
found my last patent a lot faster than I would have otherwise.
It's Ray Deshays from Caltech, who has done excellent work in tech transfer and made himself and Caltech and others happy. Okay, 30 seconds.
I myself deal with insurance for biotech companies and cyber privacy media insurance, but I think the real gap here is the angel, early, and zero stage. Because everything's so small, it's hard for institutional investors or even semi-institutional investors to put money into something that's going to get them from 50,000 or 100,000 or 500,000 to an amount that'll make sense for them. It's just so time intensive, and the returns are just empirically much larger when you start investing at the 20, 30 million stage rather than at the 50,000, 500,000 stage. I don't see ways to bridge it. Have you thought of anything?
Okay, David. Final thought.
Joel was talking about starting up a small company, and I've had the occasion to start up a couple of them recently. And the first thing I've done is to find a CEO. Because I shouldn't be out there raising the money, as somebody quite rightly said about Joel. But we really do need thoughtful managerial people who know money, who have, in the best of circumstances, already a network of people who can provide angel funding or lower level venture capital funding to get things going. I think that partnership between scientists and potential CEOs or CEOs is a good way to alleviate that, and then they can find those pockets of small money that can make a difference. And I've done that in the last couple of years, and we managed to put together, well, our first clinical trial, which will start next month, on the basis of just that kind of funding.
Okay, so I think this has been a really terrific discussion. We should come to a close here, and I want to maybe just close by thanking our panelists for participating and the audience also for participating. Thank you.
Nice job, Steve.